Hi everyone, my name is Lawore Damilola, a graduate student in the Department of Chemistry here in WIU. My mentor is Dr. Meta Sondergaard, and the topic for my research is antiproliferative effects of morel mushroom, Morchella esculenta. The aim of this study is to analyze the antiproliferative effects of morel mushroom on human ovarian adenocarcinoma cells, SCOR3 cells. Cancer. Cancer is one of the leading causes of mortality worldwide, and in 2018, it accounted for 9.6 million global deaths. So, what is cancer? Cancer is a lethal disease that is as a result of mutation in tumor suppressor genes, oncogenes, and DNA repair genes. They are characterized by abnormal cell growth and they metastasize to other parts of the body via lymphatic vessels and blood. As we can see over here, we have the primary tumor sites and we are like the lymphatic vessels. They are moved. There's organ metastases to bones, to brain, to lungs, and any part of the body. For the sake of this study, our focus is on ovarian cancer. Ovarian cancer is a form of cancer that affects the ovary and closed organs in the female gynecology. The ovary comprises of three types of cells that can develop into different types of tumors. The epithelial cells. The epithelial cells are cells that make up the outer surface of the ovary, and most ovarian tumors are epithelial tumors. We have the stroma cells. The stroma cells, they produce the female hormones, which are the estrogen, progesterone, and they also serve as structural tissues that hold the ovary together. So the stroma tumors can form from the stroma cells. And we have the germ cells as well. The germ cells produce the ova, the egg, and germ tumors can form. Ovarian cancer are malignant tumors because they metastasize to other parts of the body. From what we can see over here, we can see that there's tumor in the ovary and it is spread to the fallopian tube, to the other ovary, all the way to the GI tract, and we can also see that there's cancer in the peritoneum. What makes ovarian cancer such an eyesore? Ovarian cancer accounts for more deaths than any other cancer of the female reproductive system. It ranks fifth in cancer deaths among women. And why is this? This is because symptoms are diagnosed in late stages. Some of the symptoms include, but they are not limited to, vaginal bleeding, pain in the pelvic area, abdominal pain, and bloating. As we can see from the symptoms that has been listed, they can also be associated to some other female gynecological issues, so which makes diagnosis of ovarian cancer quite hard. We can see over here that in the first stage, which is when the tumors form in the ovary, you can see them, they are in single forms, just one, and at this point they evade apoptosis. Apoptosis is the programmed cell death. When they evade it, the body is no longer functioning properly and they progress to the stage 2. They become spheroids and it's a transitory phase. They undergo adaptive changes where those singular tumors cluster together, and when they cluster together they begin to form masses, and the third stage is moving to another site, as we have seen in pictures, diagrams in other slides where they moved from their primary site to other site. As we can see here, it first started all over here in the ovary, and we can see that it has moved to another part of the body. The general idea of cancer is mutation in a gene. But when it comes to ovarian cancer, studies have shown that mutations in breast cancer susceptibility genes 1 and 2, BRCA1 and 2, increase the ovarian cancer risk. Yes, there have been treatment of ovarian cancer like chemotherapy, targeted therapy, and genetic therapies to combat cancer, but none has been able to eradicate it effectively. Thus, the mortality rate is still on the increase. In most cases, the cancer cells, they develop resistance to the treatment regimen, leading to a relapse. This has led to the ongoing search for the cure for ovarian cancer, and this has led us to medicinal plants, which oftentimes have natural compounds. Natural compounds are products derived from plants. Research is being carried out on natural products as a possible cure for cancer. This is because many known medicinal plants are being studied to know if they have antioxidants and anti-cancer compounds. Antioxidants, because studies have shown that when people have cancer, there is an increase of reactive oxygen species and reactive nitrogen species, which is bad for the body. So if any known medicinal plants is known to have antioxidant and anti-cancer compounds, it can aid the therapy by improving the clinical prognosis. One of such is paclitaxel, which was discovered in 1971 by Dr. Wani, Dr. Taylor, Dr. Wall, Dr. Cogan, and Dr. Mathel. It belongs to the Taxus brevifolia family, and it is generally known as Pacific yew. This is the chemical structure of paclitaxel over here in the box here, and paclitaxel is commercially marketed as Taxol, and it is used in the treatment of cervical cancer, breast cancer, lung cancer, and even in pancreatic cancer. The plant of interest in this study is morel mushrooms, Morchella esculenta. It is a member of the Morchella genus. It is a highly priced edible mushroom found all over the world, but widely distributed in temperate regions like Japan, China, Korea, and Europe, and it's also found in Illinois as well. M. esculenta comes in the yellow and gray type. We can see the yellow one over here in the box below, and above we can see the gray type. Morel mushroom is of interest in this study because previous studies have shown that it has potent anti-angiogenic activities and cytotoxicity to cancer cells, and this shows a great potential to be used in cancer prevention and treatment. Also, the extracts from fruiting bodies and also the mycelium reduced cell viability in different human lung adenocarcinoma cell lines. This was done by inducing apoptosis. Finally, its studies have shown that it inhibited proliferation of human cervical carcinoma, colorectal adenocarcinoma, and hepatocellular carcinoma cell lines. With this being said, morel mushroom is worthy to be explored. Wild yellow and gray Morchella esculenta fruiting bodies were harvested from Illinois in the spring of 2019. It was freeze-dried and then ground into powder. Right on the left is the wild gray powder, and on the right is the wild yellow powder. The plant materials, which are the powders, were extracted thrice using ethanol, methanol, and water for 24 hours. Extracts were then evaporated at 50 degrees Celsius, and they were resuspended in dimethyl sulfoxide (DMSO) to a final concentration of 20 mg per ml. The extracts was filtered through a 0.22 micrometer sterile syringe filter, and the extracts were stored at -20 degrees Celsius. They were extracted thrice to make sure that there was optimum extraction. The first assay carried out is the tetrazolium reduction assay, MTT, which is also known as a cell viability assay. MTT is a colorimetric assay which measures cell metabolic activity. Over here is the MTT structure, which is yellow in color. Then in the presence of living cells, it is reduced to formazone, which is purple in color, and it is read at an absorbance of 570 nanometer. The SCOF3 cells, the human ovarian adenocarcinoma cells, were grown to 80% confluency in 50 microgram per meter gentamicin in McCoy's 5A medium. The grown SCOF3 cells were treated with extract, paclitaxel, and DMSO. Paclitaxel being the positive control, DMSA the negative control, and it was incubated for 48 hours at 37 degrees Celsius and 5% carbon dioxide. After the 48-hour incubation period, the MTT solution was added, which made the solution to turn yellow, and was further incubated for 4 hours. After the 4-hour incubation period, the formed formadone crystals were solubilized in DMSO, and it was read at an absorbance of 570 nanometer. The next assay that was carried out was the propydium iodide assay. This assay was carried out in order to assess the cytotoxic effect of morel mushroom on SCOF3 cells. The human ovarian adenocarcinoma, the SCOF3 cells, were grown to 60% confluency, and the cells were treated with water, methanol, and ethanol extracts of morel mushrooms, and it was incubated for 24 hours. After which the cells were fixed with methanol acetone of ratio 1:1. The propydium iodide is a fluorophore that binds to the DNA of apoptotic cells, which means cells that are dying. And when they are added, there will be a fluorescence that is produced. This fluorescence was then measured using the fluorescent microscope. The results for the MTT assay, each extract was statistically analyzed using one way and over the Craft-Bad Prism software and compared to DMSO. It showed that the ethanol, methanol, and water extracts of both the yellow and the gray morel mushrooms were significant when compared to DMSO, with p-values less than 0.0001. As we can see over here, we can see the 4 stars signify p-values less than 0.001 for each. All were significant. The result of the propydium iodide assay from the fluorescent microscope, the first one being the MSO, we can see that there is no much fluorescence, and paclitaxel we can see, but for all the other extracts we can see great fluorescence according to the fluorescent microscope results. Fluorescence was measured using the ImageJ software, and the measurements gotten were statistically analyzed using the one way and over again. And from what was gotten, it was seen that all the extracts were once again significant when compared to DMSO, with p-values less than 0.01 for 2 stars, and for 3 stars, p-values less than 0.001, while for 4 stars, p-values less than 0.0001. Based on the results gotten from the MTT assay and the propydium iodide assay, all the extracts for the wild yellow and gray morels showed significance when compared to DMSO, with the water extracts showing the greatest significance. This means that the Morchella esculenta has antiproliferative effects on the human ovarian adenocarcinoma cells. The future direction will be to carry out MT-TIC50 to know the half maximal inhibitory concentration of the extracts. Special thanks to my mentor, Dr. Mentor, my colleagues, the Department of Chemistry, Western Illinois University, and my family and friends. These are my references. If you have any questions, kindly drop them in the discussion and I will reply them. Thank you very much.